Jumping into atherosclerosis, and we're going to look at the pathogenesis as well as the mechanism of action. And these are really important basic foundational understandings of the pathology that and the physiology and pathophysiology of what is happening in the blood vessels that we can fix and influence. Because if we understand the mechanism of action, then we're able to better initiate lifestyle and dietary changes, support with supplements, because we have to reverse it. That's one of the best ways that we can make an impact on conservative care models of some of these metabolic diseases. So taking a look at what are the initial events in the atherosclerosis and what are causes of this. So there was a time where they thought that, the pipes, hence the vasculature, just got clogged. And we now know that it's not that simple. So endothelial injury is one of the primary initiating events. So again, you don't just wake up one day and have diabetes, and you also don't wake up one day and have atherosclerosis. These metabolic disturbances took months and months and months and years to accumulate and worsen over time. And then it becomes a feed forward negative cascade, because once it's set in motion, of course, it just worsens. And then you reach in a point where it becomes more and more symptomatic and or more disease presents. So what we want to recognize first and foremost is endothelial injury. Now remember, the endothelial cell is that single layer that's inside the blood vessels. That's your endothelium. And of course, the endothelial cell is each individual one. And when we are going to see what causes in the initial stages of endothelial cell injury, these are the primary sources. So if we're worried, where does endothelial injury come from? Then we want to help understand what we can do to positively make sure that these things don't happen. And what we also want to do is make sure that we are removing these things from the system so that we can either prevent endothelial injury and or reverse it. So what are the sources of endothelial injury? The pathological initial event of the atherosclerosis mechanism, pathogenesis, excuse me. So we are looking at mechanical stresses, hemodynamic forces, the immune complex deposits, any irradiation chemicals. And all of these can be happening. Now, many times when we have a system of hypertension and oxidative stress and a lot of our metabolic imbalances, hyperglycemia, hyperlipidemia, hyperhomocysteine, hypertension, these are going to primarily induce hemodynamic forces and mechanical stresses, especially when we're looking at the metabolic disturbance, obesity epidemic, I just blanked. MET-X, metabolic syndrome. So what we are finding is these are very prominent. However, of course, irradiation and chemicals, that could be an exposure or occupational hazard, and immune complex deposits. Well, this is a lot of our autoimmune people. So those are a few other etiologies. However, I do want you to recognize all four as being things that we have to think about when we're worried about endothelial injury in our patients. So what three things happen during dysfunctional endothelial cells responding to stresses and injury? So it's very common to recognize that in a dysfunctional endothelial cell, we're going to have increased endothelial permeability, enhanced leukocyte adhesion, and altered gene expression. And the altered gene expression, remember, everything on an epigenetic level can be turned on or turned off. So if something is overstimulated and over activated, then we're going to have that epigenetic upregulation. Just like when we have nuclear factor kappa B, that's a gene expression, and then it goes and has a upregulation of the genes that it is responsible for. And of course, that is going to be altered gene expression. And we're going to learn what an adhesion molecule is. And we're going to learn that that is one of the negative impacts that the nuclear factor kappa B being overstimulated causes is altered gene expression of overexpression of our adhesion molecules. One more event that we will learn that occurs during pathogenesis of our atherosclerosis. How does overstimulation of nuclear factor kappa B and excessive reactive oxygen species and free radicals correlate to the endothelial dysfunction? And how about our cytokines? So the most important thing to recognize is that nuclear factor kappa B can be everything in our body has a physiological role. It's when things become excessive and persistent and chronic that we have a problem. So nuclear factor kappa B is a response to a change in the shear stress on a blood vessel's wall. So once that changes by pressure and or the flow, we're going to learn that there's laminar flow, which is straight flow, and then there's turbulent flow, which is bashing and bashing and bashing up against the wall, and that's not good. So when we have that constant persistent turbulent flow, that activates the mechanotransduction signaling, and then that triggers our nuclear factor kappa B and inflammatory response. When you overstimulate it, you're going to have an overexpression of our adhesion molecules. We learn those in a few more slides, our ICAMs, selectins, and we're also going to learn metalloproteinases. And then the nuclear factor kappa B has a direct correlation to overexpression of our pro-inflammatory cytokines, especially interleukin 1, 6, here it says 12, and our tumor necrosis factor, TNF. So simply having hypertension and the loss of laminar flow is going to directly overstimulate and overactivate nuclear factor kappa B, and it's going to result in a system of oxidative stress and excessive endothelial present cytokines, inflammatory interleukin 1, 6, tumor necrosis factor, and then of course that is all going to be part of our endothelial dysfunction. And endothelial dysfunction is going back to when we have injury of the endothelial cell and we have that increased permeability, because we do not want the immune system entering excessively into the tissue. We don't want excessive leukocyte adhesion, because they'll stop and roll and then they cross over. Remember, the word diapedesis is when white blood cells roll along the tissues and then enter in between. And then of course, we already said the altered gene expression. So recognizing endothelial dysfunction is going to be present with increased endothelial permeability, enhanced leukocyte adhesion, and then of course that aberrant gene expression. And that directly correlates to overstimulation of your nuclear factor kappa B. And then that's a feed forward cycle of more and more pro-inflammatory cytokines and more reactive oxygen species. So let's see what the picture shows us for the demonstration of flow. In a healthy, normal physiological blood vessel, we have laminar flow, and it's going to be direct. And in the middle, there's a little bit difference of a vector than on the outsides. However, it flows straight. Okay, once you lose laminar flow, the loss of laminar flow now becomes turbulent flow, and turbulent flow is bashing and bashing and bashing and bashing. So what ends up happening is you have a higher mechanical shear stress on the blood vessel. So when we are looking at one of the causes of our endothelial injury was the hemodynamic force, which is the loss of laminar flow, and then the mechanical stresses, which is that shear stress from turbulent flow that is not corrected back to laminar. Now, so the question is, if we know that the body is smart, remember, our body is absolutely brilliant, and it knows how to build into the genome, how to fix and deal with these things that happen on a normal daily basis. Individuals who do not have hypertension and do not have atherosclerosis and do not have a body habitus of vascular reactive oxygen species and oxidative stress, they have episodes of vasodilation, which is the tone of the blood vessel relaxing and allowing flow to go, and then episodes of constriction. Vasoconstriction is when the tone tightens and contracts, and then that's going to influence the flow and the resistance. So what happens when the body has a loss of laminar flow, which is very typically at some of the branch points and where there are changes, or if the body through catecholamines wants to restrict blood area to a segment and shunt it to a different body area, then that would be a normal physiological vasoconstriction to shunt blood. However, when we are talking about just basic blood flow in the form of hemodynamics, we have to understand that on a regular basis, the body decides when to do laminar versus turbulent flow. And if turbulent flow happens, what we do is we initiate vasodilation. How do we do vasodilation? So if all of a sudden you have a shear stress because laminar flow has become turbulent, your blood vessel mechanoreceptors have this stimulus of increased shear stress, and that tells the body through a communication feed forward cycle that you need to vasodilate. So you relax the tone, you allow the blood flow to correct itself, and then it will simply rebound back to a normal physiological state. So it doesn't stay vasodilated, but that's the response to correct the loss of laminar flow, which is the onset of turbulent flow. So if we know we need to vasodilate and we know that the mechanoreceptors are responding and nuclear factor kappa B is being activated, what is the actual vasodilation response? And that is right here. This is NOS, nitric oxide synthase coupling. So what happens if it becomes persistent? Turbulent flow will become, it will cause endothelial injury, and if that happens long enough, you get the endothelial dysfunction. So turbulent flow directly can be an initiating event in atherosclerosis if you have the inability to vasodilate. So what is eNOS and what is NO? So eNOS is endothelial nitric oxide synthase, and NO, of course, is our gaseous vasodilator molecule, nitric oxide. And in order for eNOS, the enzyme endothelial nitric oxide synthase, to make the gaseous molecule nitric oxide, we have to have the amino acid arginine and fully reduced BH4. If you remember BH4 from diet and nutrition, that is our tetrahydrobiopterin. Tetra meaning 4, hydro meaning hydrogens on a biopterin. If you don't have equal amounts of arginine plus fully reduced BH4, you end up with a very potent reactive nitrogen species, which is peroxynitrite, and we'll learn that in a minute. So precursor arginine, cofactor fully reduced BH4. And then what happens to the cofactor with ROS? The problem with tetrahydrobiopterin is it's easily oxidized. If it gets oxidized to a BH2, it cannot serve its job. So therefore, in a system of oxidative stress, we will lose the bioavailable cofactor. Okay, so in a system that is totally functioning normal, we have NOS coupling. That is when there is enough arginine, there is bioavailable, fully reduced tetrahydrobiopterin as BH4, and we can immediately synthesize nitric oxide in response to turbulent flow. We vasodilate, we correct the flow back to laminar, and then the system will go back to normal. Now, what ends up happening in people that cannot do this is, of course, persistent turbulent flow. Once it becomes persistent, now you've got a problem. Not is that only hypertension and endothelial injury and endothelial dysfunction with the ability to increase the permeability, which we don't want, and increase the adhesion of leukocytes, which we don't want, and the upregulation of nuclear factor kappa B and the adhesion molecules, which we definitely don't want. And all of that happens if you cannot generate nitric oxide with arginine and BH4. So why do you think we supplement with arginine and citrulline for people that are having hypertension? It's because it supports the generation of nitric oxide. However, if you do not dually pair that with your antioxidants and the vitamins and minerals that support your intrinsic antioxidant enzyme systems, your mineral dependent enzymes, and your solo antioxidant reducing agents, which we reviewed coming into this segment, then you are doing an uphill battle. If you take arginine and you take citrulline and you form nitric oxide, but you don't fix the oxidative stress, all you're going to do is run out of BH4. So you can't just give arginine. You've got to also remove the excessive oxidative stress, because that is what changes the bioavailability of your BH4. You have to do both. Antioxidants protect BH4 to make sure that BH4 is bioavailable. Then your arginine and citrulline supplement is delivering the precursor, and then your body can make nitric oxide to protect itself against some of these changes. So vasodilation, your blood vessel tone is relaxing. Vasoconstriction, blood vessel tone is tightening. Which one negatively influences blood pressure? I hope you will know that that's vasoconstriction. You cannot stay in a vasoconstricted state long term, because that will induce and or worsen any hypertension and rises in blood pressure. All right, which one do we want? We want both, but we need the ability to have the vasodilation to correct for vasoconstriction and turbulent flow. Which one correlates to physiological levels of eNOS with BH4 and arginine? That's your vasodilation. That's exactly what eNOS, arginine, and BH4 generating nitric oxide does, is it's vasodilation, because nitric oxide is a gaseous vasodilator. And then what happened, which correlates to impaired bioavailability? That is your body habitus of oxidative stress and inefficient antioxidant protection. And that is going to be vasoconstriction. Now, once you have that initial response to turbulent flow, look at this. Not only do we have eNOS, which is generating nitric oxide, but check this out. I hope you recognize SOD and glutathione peroxidase. So what happens, refer back, Clinical Pearl, NRF2. What is NRF2? NRF2 is your anti-inflammatory transcription factor, and when it gets activated by oxidative stress and free radicals, it turns on the genome to synthesize and increase the expression of superoxide dismutase, catalase, and your glutathione peroxidase. Guess what? If you don't have glutathione, which is what? Cysteine, glycine, and glutamate. Cysteine, glycine, and glutamate. Guess what mineral you need? Selenium. And SOD, I hope you remember, that's your copper, zinc in the cytosol, and that is your manganese in the mitochondria. So what if a person is deficient in vitamins and minerals? They get the signal to express the antioxidants to protect against this vasoconstriction, turbulent flow, and they can't. And what about the ability to make nitric oxide? What if they have a system of oxidative stress, they don't have the ability to protect against it? Guess what they don't have? They don't have BH4. Guess what they don't get? They don't get vasodilation. So a normal healthy individual that has all of these nutrient bioavailable constituents as precursors and cofactors can respond to vasoconstriction and turbulent flow with vasodilation. In healthy, normal individuals, an episode of turbulent flow does not cause injury, because you can vasodilate and then you can protect yourself against the free radicals that are generated while the tissue is responding. If you have oxidative stress and free radicals generated in a system of sufficient antioxidant protection. You have the free radicals, but you do not have the physiological damaging effects. You have a problem with oxidative stress, if you recall, is the definition when there are more free radicals and not enough antioxidant defense protection. So the problem is the individual that cannot do one, two, or both of these. Almost always you got to be thinking both, because in a system of oxidative stress, you oxidize your BH4 and you can't make your nitric oxide anyways. So to answer the question, where I was over here somewhere, was what happens if it becomes persistent? Then it's going to correlate with the inability to couple, and we'll learn that that's called uncoupling. And then we're going to get the stimulation of nuclear factor kappa B. Now, we're also going to recognize that with the excessive expression of the adhesion molecules and the nuclear factor kappa B, that by itself starts its own cascade of more and more chemokine and leukocyte attraction. And the sheer stress alone is going to be enough to stimulate the nuclear factor kappa B. And you get your cytokines. Now, this is the next concept. I want you to be sure you recognize this. Yes, we learned about nuclear factor kappa B. Yes, we know it's the pro-inflammatory transcription factor. Yes, we know overstimulation, overexpression increases the amount of pro-inflammatory cytokines. This is the next step for you to add to that foundational understanding of what nuclear factor kappa B does as a transcription factor, and it is right here. It not only increases the expression of the pro-inflammatory enzymes that increase the synthesis of the pro-inflammatory cytokines. We already knew that. However, it also controls the adhesion molecules. The adhesion molecules play very specific roles of blood vessel reaction, and we will learn what they are in just a minute. However, excuse me, we do not want them overexpressed. Overexpression is going to equal atherosclerosis. So if we don't want our adhesion molecules overexpressed, then we do not want nuclear factor kappa B being overexpressed, because nuclear factor kappa B at the level of the genome not only controls the cytokines, new thought, I'm saying this again, the nuclear factor kappa B also, as a transcription factor, controls the adhesion molecules' rate of synthesis. If you turn on nuclear factor kappa B in your blood vessel, you turn on the overexpression of your adhesion molecules. So I definitely want you recognizing nuclear factor kappa B in the atherosclerotic model is not only excessive cytokines, tumor necrosis factor, interleukin 1, interleukin 6, and a bunch more, but it also turns on overexpression of your adhesion molecules. Okay, so what does the nitric oxide gaseous signaling molecule do? It vasodilates and it responds to turbulent flow. What happens to the tone of the muscle? It improves, and to blood pressure, it would decrease. How does eNOS, arginine, BH4, and nitric oxide resolve turbulent flow? When this is working, how does it protect the blood vessels from damage, and does it prevent endothelial dysfunction? Definitely. That's the point. Normal, healthy, optimally functioning people do not have a detrimental effect. This is how we solve turbulent flow. And when it is working, we absolutely protect ourselves and we prevent endothelial injury and we prevent endothelial dysfunction. And if you have a system where you already has endothelial injury and endothelial dysfunction, guess what? You restore eNOS and you restore vasodilation and you reverse endothelial dysfunction. They have shown over and over in medical models that this is reversible. You have to go back to the pathogenesis of why did it happen, and then you fix it. Find the trigger and remove it. Find the imbalance and fix it. And right here is what you do. It's one of the easiest ways to get people off blood pressure meds and protect their blood vessels against atherosclerosis, and to reverse it if it's already happening. So can it protect against the onset of athero? So if we know that the initial step is endothelial injury, which leads to endothelial dysfunction, which leads to hyperpermeability of the endothelial cells with diapedesis and leukocyte adhesion and an overexpression of adhesion molecules, which all cause atherosclerosis. So don't you think that if you can prevent all of that from happening by not allowing endothelial injury in the first place, don't you think that'd be a really great way to protect the population of people? I think it would. Why has it got to be so simple, and why does the world have to make it so hard? All right, what is NOS coupling? Do we want this? Yes, we do. NOS coupling is what we just looked at when we have the proper functioning of the NOS combination synthesizing nitric oxide. Now, when we get into, I know I have a slide, I'm bumping around here. When we have the opposite, which is NOS uncoupling, enos uncoupling, then we're going to have the uncoupling is exactly what it sounds like. That's when you do not have BH4 and or arginine. And what you do not get is nitric oxide. So NOS uncoupling is the system where you do not form your nitric oxide. So turbulent flow is going to activate enos, and then we're going to need to have the endothelial nitric oxide working. Where's my slide? Okay, so there's the coupling. Uncoupling is when it doesn't happen. It's in here somewhere. Okay, so let's take a look at, let's see, where am I? Do we want uncoupling? No, that is the inability to vasodilate. And then when does this happen? That happens when you don't have arginine, you don't have reduced BH4, and that happens in a system of oxidative stress because it oxidizes and depletes the bioavailable BH4. It also happens with somebody who is not eating enough protein because arginine is a protein. So you definitely need both your precursor and your cofactor. If there is ROS and it perturbs the enos cofactor bioavailability, what is the result? The result is NOS uncoupling. NOS uncoupling is the result if you have a system of oxidative stress and it aggravates by oxidizing your BH4 as your cofactor. The result is vasoconstriction, and it also generates an insane amount of peroxynitrite, and it is a perpetuating cycle of walking disease. Do we want this? I hope you know that answer would be no. How does long-term unresolved turbulent flow correlate to endothelial dysfunction, NOS uncoupling, and athero? So I hope you see that I'm just continuing to ask you the same question as you drive home the understanding that all of these correlate symbiotically and simultaneously. If you have endothelial dysfunction, you have NOS uncoupling. NOS uncoupling causes endothelial dysfunction. The process of atherosclerosis is the mechanism of action where NOS uncoupling and endothelial dysfunction coexist, and then you end up with atherosclerosis. So these are all happening simultaneously from the mechanisms that we've looked at thus far. What is the physiological response to vasoconstriction and turbulent flow, and can this happen without injury? So we already looked at it, and that is yes. As long as you have nitric oxide being synthesized and you have the ability to turn on your SOD, superoxide dismutase, and your glutathione peroxidase, which is basically antioxidant protection of the free radicals, along with the ability to vasodilate, then yes, absolutely. And you can have this occur without ever having endothelial injury. What if someone already has oxidative stress? Can they fight ROS? If they are not actively replenishing all of their essential vitamins, minerals, amino acids, and essential fatty acids, and doing antioxidants and working on reversing their hyperglycemia and getting rid of their MET-X and reversing insulin resistance, then they're just going to get worse and worse. Every one of these causes oxidative stress and sets up a body habitus of reactive oxygen species. The cool thing is each one individually does it. So when you pair these all together, it's a bombardment of ROS and RNS. So in diabetes, they demonstrated that glutathione was undetectable in the person. Undetectable. Do you think that is healthy? Absolutely not. Do you have antioxidant protection? No. Are you susceptible to free radical damage? Absolutely. So what else did they find? They found that SOD, superoxide dismutase, was what? Undetectable. Undetectable. They had no protection. They overwhelmed their physiological antioxidant protective systems because of the excessive amount of reactive oxygen and reactive nitrogen species that the imbalances from metabolic disturbances cause. So when you're treating this person, you need the basal amount of help, and then you need a whole crap ton more. They need to be super front loaded on every type of antioxidant, phytogreens, supplements, dietary fruits, and alkalizing phytogreens and produce. While they cut down on the pro-inflammatory foods, you've got to hit it from every direction. Critical thinking. So if someone already has a metabolic disturbance, excessive oxidative stress, and then you add in turbulent flow, what do you think is happening to their blood vessels? And can they protect? No. They're going to have worsening, increasing blood pressure and aggravated hypertension. They're going to have persistent chronic injury to the endothelial cells, and they will be walking atherosclerosis pathogenesis. What happens with chronic persistent NOS uncoupling vasoconstriction? How is nuclear factor involved? And let's see slide 24. So we've already looked at this, and this is, you can see that I'm just on a cycle of repeat with you here. So you can't have chronic persistent uncoupling. That is walking hypertension, that is walking ROS, RNS, and that is atherosclerosis. And that's going to be worse NADPH oxidase overexpression. That's going to be worse nuclear factor kappa B overexpression. And all of that is going to be excessive cytokines, excessive adhesion molecules. All right, so finally, I've said adhesion molecules a lot. Let's take a look at what they are. There are 3. Each one has its own function, and then they all get to play in the game of atherosclerosis. So adhesion molecules serve a normal physiological role. When they are overstimulated, overexpressed in the body habitus of excessive amounts of inflammation and excessive ROS and nuclear factor kappa B and metabolic disturbances, then they are bad. Remember, everything at a basal physiological level has a physiological purpose. It's when you overdo it with an epigenetic aberrant stimulation of the genome under a pathological body habitus that it becomes a disease. All right, so the 3 groups that we have to recognize of our adhesion molecules is ICAM and VCAM, and that's going to be those are the little arms. I picture, you remember when we were kids, we had that green rubber hand that you could smack on the wall, and it was kind of like gummy and gooey. So I picture this little gummy hand that hangs out of the endothelial cell, and it puts its gooey gummy little hand into the blood flow as everything is passing by, and it just kind of does like a little surveillance. It's like it's just like feeling around, and then it's like, okay, it's surveillance. So if you activate it, and the more and more ICAMs and VCAMs that get expressed, it's going to grab and activate the white blood cells and the monocytes. And if you grab it, you are activating it. If you have a white blood cell and or a monocyte that is simply traversing the blood, surveilling the area, and it is not activated, it is simply passing along doing surveillance. It does not become an immune response until you activate it. So if you have an adhesion molecule, ICAM and VCAM, that is sticking its little green gooey hand out into the blood as it's passing by, and it's grabbing and activating them, then that's going to start the process of the diapedesis. So the ICAM, VCAM grabs the passing by immune cells and activates them. The next group are your selectins. Your selectins are part of the transmembranes, and what they do is they allow for the rolling. They allow for the rolling of the white blood cells that were just called into the blood vessel and endothelial region, and now they're going to roll and roll and roll. And you know what happens after they roll and roll and roll on the selectins? Boom, transmigration as diapedesis. What is transmigration? Diapedesis, that is when an activated immune cell has now entered the tissue. Now, if you've got a parasite, or if you've got a foreign invader, or if you just stepped on a nail and you've got rust, you want that immune system to come in and get all that crap out of there. Do you want to activate it? Yes. Do you want diapedesis and transmigration? Absolutely. You want to heal the invader. But when you have an aberrant overexpression in your blood vessels because of injury, that is a pathological event. That is not the body protecting. It doesn't realize it's doing harm. The third category is your metalloproteinases. Many times you'll read in medical articles, MMPs, metalloproteinases. These degrade collagen. Guess what? These guys are also in your vertebral disc. And if you aggravate and overstimulate metalloproteinases, you can actually have degradation of your disc. So you might want to, if you're interested in learning about vertebral health and you want to learn how to prevent desiccation, you might want to learn about spinal vertebral metalloproteinases. Not the topic that we are talking about, but it's the same group of enzymes. All right, so if you take a look at the picture, I will do this briefly because you can do this on your own so we can keep going. Here is where you're going to have your green sticky little arm. Your ICAMs and your VCAMs are going to grab your passing by of immune cells and activate them. Here are your selectins allowing your rolling, and there's your transmigration diapedesis. And then you also have your protein matrix proteinases. Now, why is this bad if we're thinking about atherosclerosis? Why do we not want excessive metalloproteinase collagen degrading enzymes in an atherosclerotic vessel? Well, remember your atherosclerotic plug. A stable plug in an atherosclerotic vessel is less of a clot thrombus risk than one that becomes loosened and breaks off. So who's the guy breaking it off? Right here, your adhesion molecule, metalloproteinase, because it degrades collagen. So here's your vessel cap and it's going to start degrading it and degrading it. So you do not want the overstimulation of the metalloproteinases. All right, so slide 30, I directed you to. Every system and molecular service, yep, I said that, serves a physiological role. Overstimulation becomes pathological and aberrant. So you can either not let it happen, which is a prevention, or you can remove it, which is treatment, right? So that is what we do with conservative care. Find the triggers, remove them, and find the imbalance and fix it. So what B vitamin dependent molecule can become a cardiovascular disease risk when there is a deficient B9 or B12? Ooh, remember our methionine cycle. Our methionine cycle is going to recycle homocysteine with a methyl transfer of methyl B9 to methyl B12, which puts the methyl group onto homocysteine, and we regenerate methionine as we keep homocysteine from being elevated in the blood supply. So one of the most important things to know is that homocysteine directly causes the overexpression of adhesion molecules. It is one of the ways that hyperhomocysteine is a cardiovascular disease, clot, and thrombus risk. It also, hyperhomocysteine, overstimulates and activates nuclear factor kappa B. What does nuclear factor kappa B do? It goes and it turns on the genome to cause an overexpression of, yes, adhesion molecules. So homocysteine directly increases the expression of adhesion molecules, and it indirectly, secondarily increases the expression of adhesion molecules because of its activation and stimulation of nuclear factor kappa B. So what do you think we're doing in our model of atherosclerosis and hypertension metabolic disturbances, in addition to making sure that they have arginine and antioxidants, and then all of your mineral dependence, right? Copper, zinc for your SOD, you need your selenium for your glutathione, and of course you need heme iron for your catalase. But now you better be thinking B9 and B12, as well as your methyl B9 and methyl B12, which we will review next week to protect against hyperhomocysteine, to prevent and or reverse the overstimulation of nuclear factor kappa B and adhesion molecules. What are the primary contributors to oxidative stress? Slide 37. So oxidative stress comes from 1,000 and then a gazillion different sources. And the problem is these are all happening at the same time. So oxidative stress, of course, your nuclear factor kappa B. You are now expected to learn NADPH oxidase, and we'll learn a little bit more about what that is. I've already mentioned several times, be sure you recognize that your metabolic disorders equally and individually, I will say that again, equally and individually will each cause overexpression of nuclear factor kappa B. Each separately cause overactivation of NADPH oxidase, and each separately causes excessive cytokines, depletes your antioxidants, and is a system of generated, over-induced, overexpressed, over-caused ROS and RNS. It is oxidative stress. It's a body of oxidative stress. It is literally the underlying cause of every disease of the body. So hyperglycemia alone will do this. Hyperlipidemia alone, through a different mechanism, will also do this. Hypertension almost always is caused by hyperglycemia-induced hypertension and hyperglycemia-induced ROS. And NADPH oxidase, if it gets overstimulated by hyperglycemia, guess what it does? It causes hypertension. So what happens when somebody has hypertension? Oh, well, here's your first drug. Then it doesn't get better. And then a couple months later, oh, well, it's not getting any better. Here, why don't we up your dose? A couple months goes by. Then what? Oh, well, crap, still hasn't recovered. Since you already got dosed up, why don't we just add a second one in? How many standard of care prescribing primary docs have patients without any of these questions being addressed on 1 to 3 different blood pressure medications? What causes it? Oxidative stress, NADPH oxidase, hyperglycemia, and excessive nutrient intake through malonyl-CoA upregulated de novo synthesis of VLDLs as hyperlipidemia. If you have hypertension, you better go back to antioxidants, and you better go back to your hyperglycemia, and you need to start there. So oxidative stress, of course, if you don't have the ability to protect yourself from the known reducing agents, vitamin C, alpha lipoic acid, vitamin E, glutathione, as well as your intrinsic antioxidant enzymes, we've said several times, superoxide dismutase, catalase, and glutathione peroxidase. You need all of that. You don't get to choose. It's not one or the other. It's all of it working in unison in different areas and in different ways. You need all of it. That's why it's there. You need all of it. What's the other one? As I had said, NOS uncoupling is one of the worst persistent perpetuating generators of the most potent peroxynitrite, ONNO. And every time you have one round of NOS uncoupling, not only do you not get nitric oxide, you also form a superoxide anion, and you also form a peroxynitrite. The worse the peroxynitrite and the superoxide anion is in the vessel, the worse the BH4 gets oxidized, the worse the uncoupling gets. It's a feed-forward cycle. It's gnarly and it's nasty. You need to do anything and everything to make sure that it doesn't happen and reverse it. All right, so clinical Pearl, these are all occurring. It's all occurring. You have to ask, how bad is it? How many different systems? How many of these are involved? How long has it been going on? Because it's probably even worse in a dose response manner. The longer it's been going on, the more you're depleted, the more the feed forward, and the more you have to undo, and the harder you have to work at it. So what does overstimulation of the NADPH oxidase do? So let's take a look at NADPH as we get ready to talk about the foam cell. So NADPH oxidase is going to be participating in a redox reaction where NADPH is going to give away its hydrogen. However, it can, in a system where it's in excess, it will create the different free radicals. So it's protected against when it's in a normal physiological basal level generated, and there are normal physiological rates of antioxidants in its vicinity, then it is not damaging. However, when this guy gets overstimulated, which it does with oxidative stress, then it creates and it pumps out a whole bunch of free radicals. So overstimulation of NADPH oxidase is a source of extreme generated amounts of free radicals. So where is some of our oxidative stress coming from? Of course, we know that oxidative stress and free radicals and cytokines activate nuclear factor kappa B, which just makes more oxidative stress. I already said hyperglycemia, hypertension, hyperhomocysteine, all of these activate nuclear factor kappa B, and we get oxidative stress. And then our NADPH, in addition, will also be stimulated by the hyperglycemia, hypertension, and hyperhomocysteine. So guess what these metabolic disorders are doing? It is dually activating nuclear factor kappa B at the same time as overstimulating our NADPH oxidase. And the direct result is we lose BH4, and then we directly inhibit nitric oxide synthase. It directly inhibits eNOS. It directly inhibits the endothelial injury and the causes endothelial injury. So NADPH oxidase, you don't fix NADPH oxidase. You fix the sources that are causing it. You don't fix this. The NADPH oxidase is just there. Just like your nuclear factor kappa B, you want to down-regulate the rate and the amount of it being expressed by changing the different disease states and metabolic imbalances and sources that overstimulate it. So yes, overstimulation of NADPH oxidase is going to directly inhibit our eNOS. It's going to cause free radicals, which are going to oxidize our BH4. That lowers the bioavailability, and we have a worsening system where we cannot vasodilate. We have increasing blood pressure and hypertension, and that is definitely endothelial injury, which is our initial step of atherosclerosis. All right, so slides 34 through 46 are preparing you. So make sure that you recognize how all of these correlate together, and be sure you read through these and recognize it as we get into the foam cells. So let's take a look here. All right, so we know that the foam cell is going to be the macrophage activated in the intima. So we have to recall what is a macrophage. And a macrophage was a monocyte in the blood. A monocyte is going to travel, and then it gets grabbed and activated by your ICAMs and VCAMs. It's going to roll along your selectins as it goes through transmigration and diapedesis into the tissue, at which point the monocyte differentiation now becomes a macrophage. And then you have an activated macrophage. An activated macrophage is a powerhouse of cytokines, and it's a very active generating immune cell. Okay, so how does endothelial activation, chemokine chemoattraction, induce the formation of the fatty streak? So we first looked at the pathogenesis. What is pathogenesis? That is the first steps that lead up to atherosclerosis. And now looking at the mechanism of action of what is the actual foam cell, all of the events of the pathogenesis of endothelial injury, endothelial dysfunction, nitric oxide uncoupling, NADPH and nuclear factor kappa B overstimulation, overexpression of adhesion molecules all happened. All of that now brought you to boom. Now we are talking about the diapedesis and the transmigration. And once you're transmigration, once you're inside the intima, you are now the atherosclerotic foam cell. That's what the fatty streak is. The fatty streak is the accumulation of oxidized LDLs inside of activated macrophages, which are the foam cells. So let's take a look at it step by step. So all of this endothelial activation, chemokine, chemo attraction, this is what's happening with the activated endothelial injury and endothelial dysfunction. So once this is going to happen, it now calls in all of the immune cells. You get your diapedesis, and then you have your macrophage conversion and uptake of the modified LDL, and then you have your foam cell. So how does this correlate? Because that's endothelial activation and chemokine chemo attraction is exactly what's happening. In case you don't remember, just to make sure, you have a cytokine such as your interleukin 1, interleukin 6 tumor necrosis factor. And then you also have very similar other small proteins that are communication proteins, and they are called chemokines, and they specifically call more and more immune cells to an area. It's like a SOS signal that will call in and attract more and more immune cells to an area. Again, if you stepped on a rusty nail and you're about to get tetanus, you want chemokine in your blood vessel. That's not supposed to have all of this activation and chemo attraction. That is not good. So this is a pathological chemokine chemo attraction. All right. Do we want excessive recruitment and monocyte transmigration? Absolutely not. You do not want activated, overstimulated immune cells anywhere. We're talking about blood vessels, but you do not want chronically overstimulated and chronic activation of the immune system. It's just not a healthy way to live. If we don't want that, then do we want overstimulated adhesion molecules? No. You should do everything that I taught you that causes upregulation, overstimulation of adhesion molecules is what you do for treatment, and you address all of those things and you remove those things to prevent further stimulation of adhesion molecules. Do we want excessive nuclear factor kappa B, ROS, and NADPH oxidase stimulation? No. No, it's walking disease. That is walking disease. Everything that you learn about what causes these is what you change and what you remove to prevent it from happening and to reverse a body that already has it. Clinical Pearl. If all of those are etiologies for the onset and development of a fatty streak, shouldn't we avoid and remove them? Wow. Doesn't it seem like it's not rocket science anymore? Doesn't that almost seem like a duh? Right? Of course, that's what we want to do. So I hope now, like, the logic is of why we learn pathogenesis, to appreciate what it took to get to the mechanism of action of the foam cell. That's why we started all the way back at reviewing eicosanoids and dietary PUFAs and what oxidative stress is and what our antioxidants were and what our mineral dependent intrinsic antioxidant enzymes are. You had to learn that to get here. Then we had to learn what goes wrong with diabetes and insulin resistance, and how diabetes, hyperglycemia, insulin resistance upregulates the de novo synthesis that overexpresses the release of LDLs into a body habitus of a vessel that becomes excessive LDLs. And under a system of oxidative stress, you are going to oxidize those LDLs. So you don't want more of them. And then remember, insulin resistance is going to cleave and cleave and cleave and cleave the adipocyte fat, releasing free fatty acids into a system of already existing hyperglycemia, already upregulated hepatic de novo synthesis of fatty acid tags and palmitates, and it just drops the free fatty acids back into the blood. Now we have hyperlipidemia, hypertriacylglyceremia, and all of this is happening with more and more oxidative stress, less and less antioxidant protection. No, you don't wake up one day and just have diabetes. You don't just wake up one day and have atherosclerosis. You had to earn it. You did this to yourself. And now you have the opportunity to change your diet, change your lifestyle, and supplement with everything we've learned and make changes physiologically to undo all of this. And you can reverse diabetes, you can reverse insulin resistance, you can reverse metabolic syndrome, you can reverse atherosclerosis and fatty deposits, because it's all reversible. That's how beautiful this is. All right, so shouldn't you avoid and remove them? Absolutely. Yep. I tend to be tough love with my patients. I'm tough love. I can only what's saying? I can take a horse to water, but I can't make them drink, right? So the careful thing is you can't care more than them, because when I want it more than them, I get frustrated and my tough love comes up and I'm like, okay, well, if you can't not want to feel how you are and not want to do anything about it either. This is not an easy lifestyle to give up all of your favorite things, and this doesn't come with a free pill. So this is actual effort, but now at least you understand how to help people do it. All right, since it's physiologically valuable to avoid them and remove them, doesn't it help to know how? Yes, that's why I started you in reviewing all of the lectures that we did to get you to this point, and I started you in 4th try as soon as I got you under my wing. All right, clinical pearl. Anything the CBC, CMP, ferritin, B9, B12, advanced lipid tests with LDL phenotype, ApoE, lipoprotein, PLA2, and CRP, it will give us baselines. I'm so proud of you, because this week you now know what all of that means, and you know what it's telling you, and you now know how to order it, and you now know what to do with it. Very proud of you. All right, clinical pearl. Applying foundational info from the 4th try on treating nutritional deficiencies to support enzymes, cofactors, oxidative stress, and essential fatty acids, and using ketogenic low-carb diet is central to diagnosing, testing, and successfully reversing the pandemic. We will look at a couple PubMed studies. It takes about 60 days of going ketogenic and getting rid of the sugar, and you can completely reverse non-alcoholic fatty liver. You can completely reverse metabolic syndrome. The weight did not immediately come off. There was still obesity, but the obesity was present without the metabolic disease markers of metabolic disease, hyperglycemia, hyperlipidemia, insulin resistance, which are your heart attack, cardiovascular risk factors for stroke, clot, thrombus. So it's definitely reversible. All right, what does monocytes become? We said macrophages. What is macrophage differentiation? That's when you have an activated macrophage becoming a pumping out system of cytokines. Do activated macrophages express LDL receptors? And how about scavenger receptors? All right, so be sure you recognize and go back and review these on your own. You need to understand this in the stepwise manner. And if you understand it as a story, picture this happening, it won't make it memorized. You won't have to memorize it. If you understand that all of these things are happening and then each one leads to the next, this will be a lot easier for you to learn and remember. This is probably a large portion of your exam, and this is how you fix people. So I really expect you to know these. So be sure you definitely focus on that. All right. So as we get into the macrophage differentiation, now that we're looking at the mechanism of action of the fatty streak, which is your foam cell formation, we need to appreciate that your monocyte got called in by chemoattraction and by adhesion molecules. Your ICAMs and VCAMs grabbed and activated it, your selectins allowed it to roll along, and then you had transmigration and diapedesis. Once it's inside the tissue, now we're talking the macrophage, and monocyte is transmigrating and it's turning into the macrophage, and that's in the intima blood vessel. So as the macrophage differentiates, now here's answering your question. What is the macrophages expressing? If you recall, an LDL receptor is expressed on a tissue, and that allows the LDL particle ApoB100 on the LDL particle to bind to the, yes, LDL receptor. The ApoB100 on the lipoprotein LDL is expressed, and that binds to the LDL receptor on the tissue. Now remember, in normal healthy tissues that need cholesterol to either repair their plasma membrane or to use cholesterol for a synthesis pathway, it will put out an expressed LDL receptor when it needs it. A normal, healthy, physiological tissue that does not need cholesterol will not express the LDL receptor. So what are we finding here? Macrophages are expressing the LDL receptor once they are activated inside the intima, the intima blood vessel. What else is it expressing? Ooh, we're learning the scavenger receptors. The macrophage is going to express what we call as scavenger receptors. And we're going to learn what those are in just a minute. So what happens is we have a problem because the macrophage will take up modified LDL. We're going to compare that to nascent LDL. So when you have modified, that primarily is going to be your oxidized LDL, and that is a completely different molecule than nascent and LDL. Nascent LDL does not have any changes to the lipoprotein PUFAs on the outer lipoprotein phospholipid layer. Also, what do we see here? We have uninhibited, disrupted cholesterol efflux, and now we've got the foam cell. You need to recognize all of that has happened with your macrophage differentiation after the monocyte transmigration occurred. All right, so how does modified LDL enter the intima macrophages, and what happens once inside? All right, so we have to do a review of last week. So what happens with normal LDL uptake? Normal LDL uptake expresses an LDL receptor when it needs cholesterol. Your ApoB100 comes along and it binds. And then remember, endocytosis, the whole thing gets engulfed. The whole thing, the LDL particle with the ApoB100 plus the LDL receptor all gets taken up into its inside. We call that endocytosis. Okay, so this is nascent, unmodified, healthy, normal, physiological LDL. That whole complex that just got endocytosed is then going to go into the lysozyme, and the lysozyme is then going to degrade the whole thing. The entire LDL particle, all of the phospholipids on the outside, all of the cholesterol on the inside, plus the ApoB100, all gets recycled in the lysozyme. You know what else gets recycled in there? The LDL receptor. So the whole thing gets broken down. This is normal. This is called regulated LDL uptake. Then the cell uses what it needs. It stores a little bit as esterification. And then you have to remember, this is the key word, cholesterol efflux. Any extra that the cell did not need after it stored a little extra as esterification, it kicks out as cholesterol efflux, hence out, and it puts it on an HDL particle for HDL reverse cholesterol transport to take it back to the liver. What does the liver do? Primarily dumps it into bile to be released into the enterohepatobiliary system, which is the intestines. Okay, so what is the scavenger receptor then? A scavenger receptor is going to be this aberrant pathological expression of scavenger receptors. So what is a scavenger receptor? There's two of them that you have to know. The two that you have to know are going to be your CD36 and your LOX1. CD36 and your LOX1. So a scavenger receptor that gets expressed on the activated intima macrophages, which are become your foam cells, they express the two, there's more, these are the two primary ones, CD36 and LOX1. These are your unregulated, oxidized, modified LDL uptake. So what does unregulated mean? Unregulated means that unlike our normal LDL receptor, which gets endocytosed, and that stops any more LDL particles from entering the tissue. The opposite is true. Now, pay attention. When you have to know every step that I'm about to say. When you have the CD36 and the LOX1 expressed scavenger receptors, they only take up modified and oxidized LDL. They do not respond to the normal physiologically healthy LDL, nascent LDL. Now, unlike normal LDL, cholesterol regulated uptake, where endocytosis removes the LDL receptor, once this CD36 and LOX1 takes up modified LDL by taking the modified LDL and putting it into this activated macrophage foam cell, it keeps going. If there's more oxidized LDL, which of course there is, it will eat more and eat more, and the LDL keeps going inside, which is modified and oxidized. And then guess what? More and more CD36s and LOX1s get activated, and they just take up as much as they can get, and they kick it inside. Unregulated LDL uptake. So which one is physiologically normal? LDL receptor endocytosis of nascent LDL. That is a completely normal physiological process. What is pathological? Scavenger receptor 36, CD36 and LOX1. Pathological, aberrant, unregulated, disinhibited, continual, perpetual uptake of modified LDL. The scavenger receptors, pay attention, do not respond to nascent physiological normal LDL. They only uptake modified LDL, and they do it continuously. They don't stop. Now, here's the next part that you have to know. So we're still telling the story. What is this impaired efflux? What is the problem with foam cells? We are comparing normal physiological LDL response and efflux to the impaired efflux of the scavenger receptor aberrant LDL modified uptake of your foam cell. We are explaining the mechanism of action of why foam cells become foam cells, and it's right here. So, unregulated, continuous uptake of modified LDL through scavenger receptors. Guess what? They get inside. Guess who's inhibited? Your lysosomes don't work. Your foam cells have disrupted, dysfunctional lysosomes. So there's no breaking down. Then what else happens? You don't have cholesterol efflux going on to HDL. It goes in, it can't get broken down, and it can't get out. And then you have no HDL taking it back, and that is impaired efflux. There is very little HDL reverse transport happening. So all of this is happening in the foam cells. So we answered all of that. And again, I think it's easiest if you look at it as a story. Recognize the normal physiological is your target. You've got to get it back. You've got to remove the stimulus and decreasing your N6, supplying antioxidants, fix your reactive oxygen species, decrease them, get rid of as much anti-inflammatory stimulus as you can. So be sure you recognize the sequence of events and the difference between nascent and oxidized LDL. All right, so let's see here. Slide 17, the pathological uptake and retention of the oxidized and modified LDLs has what detrimental pathophysiological event on the blood vessel endothelium that induces and worsens the damage that causes the fatty streak? So I took you back to slide 17. You do not want activated endothelial cells. You do not want monocyte activation and diapedesis. So the worse this gets, the more activated endothelial cells that there are. Activated endothelial cells send out your SOS signals, and that is the source of the chemoattractants. The chemoattractants are the chemokines that call more and more and more immune cells and monocytes to the area. It is a feed forward perpetuating pathological process. And the activated endothelial cell is releasing chemokines. It's expressing excessive amounts of adhesion molecules. And the worse the foam cell gets inside, the more activated endothelial cells you get. So this is, again, this is a big feed forward cycle. All right, clinical pearl. So did someone wake up one day and all of a sudden magically have atherosclerosis? Nope, nope, doesn't work that way. Or did it take months and months and different and poor dietary and lifestyle choices to edge it along? Yep, it took effort. All right, so this is interesting. Ready? Critical thinking. So after learning the pathogenesis and the mechanism of action, reflecting back to last week's lipid panel, what is your perspective of reading a lipid panel without knowing HB1AC? That's your prediabetes marker of chronic blood hyperglycemia. How about your LDL particle size? Small, dense, atherogenic. CRP, that would be an indicator of non-specific inflammatory body habitus. So, is it cholesterol, the molecule? Is it? Or is it a body habitus of reactive oxygen species, oxidative stress, metabolic disturbances that caused it and worsened the reactive oxygen stress, that depleted your antioxidant protection, it oxidized your BH4, it induced NOS uncoupling, and then that directly caused vasoconstriction and worsened your hypertension, caused endothelial injury, which is your initiating event of the atherosclerosis pathogenesis, but also an additional psychological source of chronic and persistent, what did we say? RNS and ROS, peroxynitrite, superoxide, anions, and a crap ton of just more and more free radicals, which all lead to the event, our mechanism of action of the fatty streak, which is our foam cell, which is taking up an oxidized LDL in an unregulated manner, right? Initiating the atherogenic pathological process. And it's driven by worse and worse levels and ratios of your eicosanoid, poor dietary omega-6 diet, because that gets placed on your P2s. What happens on P2s? That is your eicosanoid generation. Your PLA2 cleaves it off. You get COX and LOX. And then you have excessive amounts of eicosanoid series 2 and 4 prostaglandins and more cytokines, and that is all in a bath of vascular ROS. Or wait, or is it just cholesterol? Okay, no, it's not. It's not cholesterol, the molecule. It is this. It is this. So, I hope I have had a very strong influence on you being able to picture and analyze and perceive the hemodynamics, the reactive oxidative stress that is occurring, and how it stems from deficient macro and micronutrient and overnutrition directly correlating to insulin resistance that disrupts the liver's functioning as far as the hepatic de novo synthesis of your VLDLs, which are a direct source of extra LDLs, which cannot be around in a body habitus of inflammatory markers. All right, so next week we will continue some of our testing. We'll do a couple case studies, and yes, my mom got her labs back, so super excited. We will cover our case study with my lovely mother's blood work. Now, of course, you don't get her name. That's a HIPAA, but super excited. Have a good weekend, and I will see you next week.